Alrighty guys, just a quick video to talk through this 14.4 section of the textbook that I didn't quite get to the other day. So the key knowledge we're looking for out of this section is the concept of the rational drug design in terms of the complementary nature, shape and charge of small molecules that are designed to bind tightly to target biomolecules limited to enzymes, resulting in the enzyme's inhibition, which that's a concept that we are already really familiar with from our work on enzymes. We know all about enzyme structure and the activity of the active site and how important that is to the enzyme's function, and how through inhibition, be it competitive or non-competitive, that active site can be altered so that it no longer binds to the substrate. And the rest of the key knowledge reads, giving rise to consequential therapeutic benefit, illustrated by the Australian development of the antiviral drug Relenza as a neuraminidase inhibitor. There is another short key knowledge section that's related to 14.4 that is just about the use of chemical agents against pathogens, including the distinction between antibiotics and antiviral drugs, with reference to their mode of action and biological effectiveness. Again, something that we are already really familiar with from previous chapters' work when we looked at pathogens and treatments against them. But looking first at the rational drug design, and you're given at the start of 14.4, you're given this little quick intro to Relenza as a concept, but it is covered a little bit better further into the chapter, so we'll go find that. So looking at our use of chemical agents against pathogens generally at the start of the chapter, and it actually does those dot points back to front. So it's talking about antibiotics first, and a really nice reminder of antibiotics' function, summarized really well in that diagram there. So that is the effectiveness of antibiotics and what they actually do to a bacterial cell to prevent them proliferating in the body. So things like inhibition of cell wall synthesis, weakening the bacteria's structure, disruption of the plasma membrane, inhibition of metabolic pathways means that it's blocking the chemical reactions that would normally be happening inside the bacteria, allowing it to grow and live. Inhibition of protein synthesis. We can hopefully have an appreciation now of why that would be such a detrimental thing to happen. Proteins are life. Bacteria also need to be able to produce those proteins in order to continue living. And inhibition of DNA or RNA synthesis. That's a really nice one to point out, because if we inhibit DNA synthesis, the bacteria can't divide. We need, for binary fission to happen, a bacteria needs to be able to make a copy of its DNA. And if that can't happen, the bacteria won't divide, the bacterial infection will cease. So antibiotics, there's a little bit of reading to do around antibiotics and their effectiveness, and then followed by antiviral drugs, which work with the virus's action or activity in mind. So in order for a virus to infect a cell, it needs to gain entry to that cell. It needs to be able to insert its genetic material into that cell, and then have the cell either incorporate the gene as part of its own DNA and perform transcription and translation, or just have the gene itself in the nucleus being read and transcribed and translated to produce more viral parts. And if we can block that activity at any one of those points, then we've got an effective antiviral drug. So they do things like preventing the virus from entering or from injecting its DNA into the cell in the first place, inhibiting enzymes that catalyze reproduction of the virus genome, so stopping those enzymes working, blocking the transcription or translation. So that's all a very broad description of how you would stop the virus at any one of those important parts. So that's that second dot point that gets knocked over first in the chapter. And then we are looking at the concept of the rational drug design. So in your OneNote, you are asked to complete this diagram here. And that diagram relates directly to this one in your textbook. So have a read through what the rational drug design is, and it is something that you've actually seen in action, although not targeting an enzyme. But we've seen the development of the COVID vaccine in real life taking place right in front of our eyes. And it follows much the same pathway. So identifying the target molecule, we are asked to look at the concept of rational drug design in terms of an enzyme that needs to be targeted. But for the development of the COVID vaccine, the molecule would have been looking at the structure of the virus and seeing if we could establish what that looked like. So determining the shape of the molecule, having a look at the drugs that already exist. Is there already one that is effective against whatever we are trying to switch on or off? Is there one that already targets that shape? Take or design a new one. That's important. Or design a new one. Take that drug, that molecule that's been either found or made, and test it on the target molecule. Does it fit? Does it effectively inhibit the enzyme? Test on animals first, so that will be the animal trials, the clinical trials, and then clinical trials for humans. There's lots of good movies and viewing that you can have a look at for stories like this. One that I've seen before is called Extraordinary Measures, starring Harrison Ford. I think I saw it as a daytime movie when I was home on maternity leave once. But it's a good movie. It's all about the targeting or the development of a drug against a genetic disorder, against an enzyme that is building up, I think, in the system of children, and leading them to this very degenerative disease. And in order to get a good outcome for them, they need to be able to get rid of this enzyme. So, rational drug design at work. Okay, but you need to have this very specific example of Relenza. So the chapter, the textbook starts talking about here on chapter 573. But it is summarized really nicely in this diagram here, which I have cropped into your OneNote, and you need to describe in your own words what's going on here. So you're first shown the case of what's happening with influenza under normal circumstances, so without the presence of Relenza. And so we can see the virus here, and this would be the host cell down here. And the virus is exploiting these receptors on the outside of the host cell and holding on to them. So this little surface protein that it has called hemagglutinin, it has a shape and the ability to grip onto the receptors on the face, on the surface of the host cell, holding the virus in close proximity and allowing it to deposit its DNA into the host cell. Once that virus has injected its DNA into the host cell, these very important enzymes here, the little scissor-shaped ones on the surface, they're called neuraminidase. So, and that's a keyword from the key knowledge. Neuraminidase inhibitors we're looking at. So this is the target enzyme. These neuraminidase, they cut the protein on the surface of the host cell and allow that virus to go off back into the body and infect more cells. So that was established. That was the first part of the rational drug design that we needed to see. What was the mode of transmission of the viral DNA? How was it making contact with the host cell? what are the shapes of these proteins that are involved? That's all the first part of the rational drug design, to work out what's going on at a molecular level first. And then the question was asked, well, can we switch off the function of this enzyme? And these are familiar shapes that we've got going on here. You can see that we've got another molecule labeled neuraminidase inhibitor, and it's got a complementary shape to this neuraminidase enzyme. And so it is the inhibitor of the neuraminidase enzyme. So what happens when Relenza is introduced to the virus? It attaches to that active site and blocks the activity of the neuraminidase. What that looks like then as part of the activity of the virus, well, the virus its hemagglutinin is still active, so it can still grip onto the host cell, and it can still do its virus thing and insert its DNA into the host cell. However, that's it. That's the end of the story. So we've now got one infected cell, but we won't have all those other cells being infected by the same virus because it can't detach itself now. The function of that neuraminidase is suppressed, and the virus can't release itself from the host cell. So we end up with a case of one host cell being infected. And of course, when a virus has gained entry to the body, it probably has already replicated. And this would be more than just this one virus. So it's greatly slowing down the effectiveness of this virus because we're only infecting one cell. That cell can still make copies of the virus, and that cell's still probably not going to fare too well. But hopefully we've got enough Relenza in the system that those new viruses that then are made or generated by this host cell can also be suppressed. So it's not completely stopping the action of the virus, but it is greatly slowing it down. All right, so hopefully that helps you with your understanding for chapter 14.4. I'll see you online.